Assalamualaikum warahmatullahi wabarakatuh. I, Nur Nazifa binti Muhammad, the first presenter that will talk about introduction of blood, artificial blood, objective of case study, and lastly is content 2. Okay, first of all, what is blood cell? Okay, blood cell is a circular thin fluid which providing body with nutrient, oxygen, and waste removal. It is made in bone marrow, and there are several components of blood, which is plasma, red blood cell, white blood cell, and platelets. All of these components have their own functions, which is the first one is red blood cell is to transport oxygen from the lung to the body's tissue and transport carbon dioxide from the tissue to the lungs. While for the white blood cell, the main function is to fight infection and aid in the immune process. And the last one is platelet for the blood clotting. Okay, next is what is artificial blood. Okay, artificial blood is also known as blood substitute or blood surrogate. Okay, it is a substance that used to mimic and fulfill some functions of the real blood. Artificial blood products have the potential to save life in emergency situations. Okay, there are two types of artificial blood substitutes that scientists are working on. The first one is perfluorocarbon emulsion, and the second one is hemoglobin-based oxygen carriers. There are three objectives in this case study, which is the first one is to determine how artificial blood is made. Second, to compare the effectiveness of artificial blood with real blood. And the last one is to determine the pros and cons of artificial blood.
Assalamualaikum warahmatullahi wabarakatuh. My name is Azina Nazira binti Azali. So I will explain about how artificial blood is made. Artificial blood can be produced in different ways using synthetic production, chemical isolation, or recombinant biochemical technology. Synthetic hemoglobin-based products are produced from hemoglobin harvest from an E. coli bacteria strain, and hemoglobin is grown in a seed tank and then fermented. The ideal artificial blood product has the following characteristic. First, it must be safe to use and compatible within the human body. This means that different blood types should not matter when the artificial blood is used. It also means the artificial blood can be processed to remove all disease-causing agents such as viruses and microorganisms. And second, it must be able to transport oxygen throughout the body and release it where it is needed. And third, it must be shelf stable. Unlike donated blood, artificial blood can be stored for over a year or more. This is in contrast to natural blood, which can only be stored for 1 month before it breaks down. And there are two significantly different products that are under development as blood substitutes, and they are different primarily in the way that they carry oxygen. One is based on PFC, while the other is a hemoglobin-based product. I suggest PFC or perfluorocarbons are biologically inert materials that can dissolve about 50 times more oxygen than blood plasma. They are relatively inexpensive to produce and can be made devoid of any biological materials. This eliminates the real possibility of spreading an infection disease via a blood transfusion. From a technological standpoint, they have two significant hurdles to overcome before they can be utilized as artificial blood. First, they are not soluble in water, which means to get them to work, they must be combined with emulsifiers, fatty compounds, called lipids. They are able to suspend tiny particles of perfluorochemicals in the blood. Second, they have the ability to carry much less oxygen than hemoglobin-based products. This means that significantly more PFC must be used, and one product of this type has been approved for use by the Federal Drug Administration (FDA), but it has not been commercially successful because the amount needed to provide a benefit is too high. Improved PFC emulsions are being developed, but have yet to reach the market. Second, hemoglobin-based product. Hemoglobin carries oxygen from the lungs to the other tissues in the body. Artificial blood based on hemoglobin takes advantage of this natural function. Unlike PFC products where dissolving is the key mechanism, oxygen covalently bonds to hemoglobin. These hemoglobin products are different from whole blood in that they are not contained in a membrane, so the problem of blood typing is eliminated. However, raw hemoglobin cannot be used because it will break down into smaller toxic compounds encompassed within the body. There are also problems with the stability of hemoglobin in a solution. The challenge in creating a hemoglobin-based artificial blood is to modify the hemoglobin molecule so these problems are resolved. Various strategies are employed to stabilize hemoglobin, and this involves either chemically cross-linking molecules or using recombinant DNA technology to produce modified proteins, just as polyethylene glycol modified liposome encapsulated hemoglobin, nanoparticle and polymer cell encapsulated hemoglobin, stabilized hemoglobin solution, polymerized hemoglobin solution, conjugate hemoglobin solutions. Conjugation of hemoglobin effectively increases its molecular size and reduces antigenicity, resulting in a slow rate of removal from the circulation and reduced visibility to the reticuloendothelial system. Unique features of conjugated hemoglobins are their high oncotic pressure, which makes them very potent, plasma volume expanders, and their viscosity. Intramolecular cross-linked hemoglobins are not significantly increased in molecular weight, but have specific chemical cross-links between polypeptide chains that prevent dissociation to dimers or monomers. These modified hemoglobins are stable and soluble in solutions. Theoretically, these modifications should result in products that have a greater ability to carry oxygen than our own red blood cells. It is anticipated that the first of these products will be available within 1 to 2 years.
Second, how effective artificial blood compared to real blood? Okay, first of all, artificial blood can avoid complications associated with real blood transfusion, such as immune reaction to the blood, second, infection risk, and then as we know that if you are using real blood to donor, the donor blood must match with the recipient's blood type. But then if you are using artificial blood, it doesn't matter because it can match any type of blood. And then human blood must be refrigerated and has a 42-day shelf life, whereas artificial blood products in development can be freeze-dried and stored at room temperature for up to 2 years. Okay, next, testing for hepatitis, HIV, and other pathogens in donor blood raise the cost of transfusion. And the last one, even though the tests are highly sophisticated, the risk of infection cannot be completely eliminated. Okay, next, as the number of cigarettes increase, so does the number of people who require blood. So with an aging population, the number of willing or able to donor blood does not match with the need of the blood. So by having artificial blood, it is a very effective way for blood donation. Okay, next, artificial blood is sterile and does not require blood type matching, just like I mentioned before this. So it could potentially save life in emergency situations or in remote locations. Okay, artificial blood could potentially deliver oxygen faster than real blood during an emergency, and this can help to minimize tissue injury. Okay, blood substitute could help people with blood disorder who require regular long-term blood transfusion, such as those with MDS, which is myelodysplastic syndrome. It is a kind of cancer that prevents immature red cells in the bone marrow from maturing. Okay, second is aplastic anemia, which is the bone marrow does not produce enough new blood cells. Last but not least, artificial blood may be used to better preserve donor organs and prevent reperfusion injury, which is organ tissue injury caused by the return of blood supply. All of this explanation shows that how effective artificial blood compared to real blood. So I think that's all from me. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Najwa binti Muhammad Ramadan, and I will continue with content 3, which is pros and cons of artificial blood. I will discuss about the pros of artificial blood. Based on the effectiveness, artificial blood do not react with oxygen and is also easy oxygen transport. Artificial blood can carry more oxygen more than normal blood. It also can increase oxygen solubility in plasma and also allows for immediate full capacity oxygen transport. Next, artificial blood also easy to handle. It can be stored at room temperature. While for the normal blood, It only can be stored at temperature 2 degrees until 6 degrees Celsius. Artificial blood also can be stored for a long period of time. Blood need to be refrigerated and last for 42 days, but the artificial blood can last up to 3 months unrefrigerated. Next is no-risk infection. It can reduce the spread of HIV and hepatitis or other blood-transmitted disease. It also no risk of infection of bacteria or germs because we can sterilize it. And then there are no antigenic molecule in artificial blood. Next, the demand for blood transfusion rising. It is because the artificial blood is universal so that everyone can use it, and the artificial blood don't need to depend on donors because it don't have the type of blood. But for normal blood, it have a type like A, B, and O. Artificial blood also can use when emergency because it doesn't have the type of blood, so it can be used for everyone. Next, the religious issue solved. This artificial blood is an alternative for those patients that refuse blood transfusion. Some of the religious cannot accept the blood transfusion, but with this artificial blood, it can accept it. And this artificial blood can be used for people with religious belief like Jehovah's Witness. Now, we move to cons of artificial blood. The artificial blood may cause a side effect. The first one is it may increase chance of heart attack. It also don't offer defense against disease, and sometimes it often cause flu-like symptoms and also cause allergic reaction. Second one, it also have a bad features. Artificial blood don't carry dissolved food. It only function on carrying oxygen, and it also don't clot blood. It is because the platelet count in our blood will decrease. It is also difficult to get into the system. The third one is the cons of artificial blood, the types of PFC emulsion. PFC can cause phagocytosis by recipient organism immune system. PFC also cannot be used in human body and must be discarded. PFC absorb oxygen passively, so that patient must breathe at linear rate to ensure oxygenation of tissues. Fourth one is the artificial blood is so expensive. It is because in order to make the artificial blood, we are lack of raw material, and it also need a lot of water. It makes the cost of making artificial blood increase. That's all. Thank you.
For conclusion, currently there are several companies working on the production of a safe and effective artificial blood substitute. The various blood substitutes all suffer from certain limitations. For example, most hemoglobin-based products last no more than 20 to 30 hours in the body. This compares to transfusion of whole blood that lasts 34 days. Also, these blood substitutes do not mimic the blood's ability to fight disease and clots. Consequently, the current artificial blood technology will be limited to short-term blood replacement applications, and in the future, it is anticipated that new materials to carry oxygen in the body will be found. Additionally, longer-lasting products should be developed, as well as products that perform the other function of blood.